So, and today's topic of discussion is GI secretion. It is the first lecture of GI physiology, and in this discussion we'll be discussing about all these things: salivary secretion, gastric secretion, we'll be talking about pancreatic secretion, similarly bile secretion, and last topic intestinal secretion. So here, before getting into all these topics, the first thing that we would like to go toward is we will look toward the anatomy, right? The anatomy of our GI system. So under GI system, what are the things that comes under GI system? So initially esophagus, right? You can see upper and lower esophageal sphincter. Similarly, we have stomach right here, right? We have stomach right here. Here you can see pylorus, right? Pylorus, the last part of stomach. Similarly, we have liver, the largest organ of human body, right? You can see gallbladder right here. Similarly, we have pancreas right here. We have what after this stomach? What we had is we have what? We have small intestine, right? We have small intestine, duodenum, ileum, and jejunum, right? That will go behind this structure. And we have colon, right? Under colon we have what? We have ascending colon, we have transverse colon, we have descending colon, we have sigmoid colon, we have rectum, and lastly we have what? Anal canal, right? It is ileocecal valve. You can see right here, ileocecal valve. What is the ileocecal valve? It is the starting point of our large intestine, or we can say the ending point of our small intestine. So here small intestine, colon, the part of large intestine, right? Here you can see the sphincter of Oddi, right? Okay, this is this is all about what? This is all about the normal GI system, the parts of normal GI system. So before getting into the real discussion, I would like to mention these things. I mean, this is so important, the histology of GI system. Talking about the different layer, right? There are two main layer. Okay, there are four main layer. The four main layer are, okay, you can see the four main layer. One is mucosa, right? One is submucosa. Another is what? Muscularis propria, and last is serosa. So you can see what is this? This is our lumen. The innermost one is our mucosa, and the outermost is our serosa. So talking about the innermost one mucosa is further divided into further layers. The epithelium covering, right, and then our basement membrane, our lamina propria, and muscularis mucosa. So the one thing that we must be conscious right here is we need to be conscious about muscularis mucosa, because we have another layer right here, that is muscularis propria. So muscularis propria is right here, but muscularis mucosa, we are talking about mucosa, right? The layer of mucosa, it is right here. Similarly, after mucosa we have submucosa, right? We have muscularis propria, and under muscularis propria what we have is we have the inner circular muscle and the outer longitudinal muscle. Similarly, between inner circular muscle and outer longitudinal muscle we have myenteric plexus, right? What is this myenteric plexus also called? This myenteric plexus is also called Auerbach plexus. What I am saying is Auerbach plexus. What is the function of this myenteric plexus? This myenteric plexus, it helps in motility. It helps to perform the function such as peristalsis, right? Okay, peristalsis. So this is the function of this myenteric plexus or Auerbach plexus, right? Okay, similarly, okay, here we said that muscularis propria have what? Myenteric plexus. Similarly, as it has myenteric plexus, our submucosa also have what? Our submucosa have Meissner's plexus. Meissner's plexus. Now, what is the function of this Meissner plexus? Meissner plexus help in secretion and absorption, right? It helps in secretion and absorption. Okay, we can just make one mnemonics. What is this? M, Meissner's plexus, and what is this? S, secretion and absorption, right? Secretion and absorption is the function of our what? Our Meissner's plexus. Similarly, what is the function of myenteric plexus? It helps in motility. It helps in peristalsis movement. That's all. Now, moving forward, okay, we said that muscularis mucosa, right? It is also a muscular layer as our muscularis propria. We said muscularis propria contains myenteric plexus or Auerbach plexus that helps in peristalsis or motility. But this muscularis mucosa has no role in peristalsis or motility, right? Motility. Okay, then what is its function? Its function is that during the time of contraction and relaxation of this muscularis mucosa, it helps in altering the surface area of our GI system. That is why, right? That is why it helps in what? Increasing or decreasing surface area for absorption or what secretion. So that is the function of muscularis mucosa. So we are clear about these things, right? The outermost one is our serosa, innermost one is our mucosa. Mucosa further divided into four different layers. The epithelial lining, basement membrane, lamina propria, right? And this lamina propria, what is this? This is the layer that consists of what? Connective tissue. Okay, and muscularis mucosa, we said that it helps in what? Its relaxation and contraction helps in altering the surface area, and that is its function. Similarly, talking about soft mucosa, it consists of what? Mesenteric plexus. What is the function of mesenteric plexus? The function of mesenteric plexus is what? Secretion and absorption. Similarly, talking about muscularis propria, it consists of inner circular muscles. How to remember? I, circular's I, right? Inner circular muscles and outer longitudinal muscles. And within inner circular and outer longitudinal, we have mesenteric plexus. Now, what is this mesenteric plexus? It is also called Auerbach plexus, that helps in peristalsis or motility. Okay, and the outermost one is our serosa. So this is all about histology of our GI system. Okay, now moving forward, okay, we are talking about GI secretion, right? We are talking about GI secretion. Now let us move to our topic, and that is GI secretion. Now talking about GI secretion, okay, talking about GI secretion, what we have is total fluid secretion in GIT is about how much liter? About 9 liter, right? Total fluid secretion in our GIT. Now out of this 9 liter, what we have is we have 8.8 liter to be absorbed, right? Absorbed. And what about 200 ml? 200 ml, right? 200 ml is excreted. What is happening? It is being excreted. Okay, it is being excreted, right? Okay, now we'll be talking about what? We'll be talking about all these 8.8 liter which is being absorbed. Okay, right? Okay, now let us move toward this diagram. Okay, in this diagram, as we have mentioned earlier, what is the total fluid secreted in GIT? It's 9 liter, right? Now what about absorbed one? Absorbed one is about 8.8 liter. Similarly, excreted in stool 200 ml, sorry, 200 ml. Now okay, now GI secretion, what GI secretion includes? GI secretion, okay, GI secretion includes our what? Saliva, right? Okay, let us begin from up, right? Okay, what we have right here, we have mouth, right? In the area of mouth, what do we have? We have saliva. Okay, saliva, right? What is, okay, saliva? In the area of mouth, we have saliva secretion. That is also under our GI secretion. Now as we move further, what we have is our food now reaches stomach, right? In stomach, what we have is we have gastric secretion, right? Now gastric secretion is the highest amount of GI secretion. It is the highest amount of GI secretion. What is its amount? Its amount is 2.5 liter per day. 2.5 liter per day. And okay, and another important fact about it is the most acidic secretion in our GI system. It is the most acidic secretion. And what is its pH? Its pH ranges from 1.5 to 3. So we talked about saliva, we talked about gastric secretion, right? Gastric's maximum amount of secretion, right? Maximum amount of secretion 2.5 per day. Most acidic 1.5 to 3 pH. Okay, now as our food has already reached stomach region, now moving downward, what we have is we have what? We have our pancreas, right? Pancreas between what? Between what? Stomach and intestine. So we have in that region we have our pancreatic secretion. Now what about pancreatic secretion? Okay, right? Okay, and talking about pancreatic secretion, we have talking about pancreas, we have exocrine and endocrine part. So here we are not talking about endocrine part, because in for the digestion of food, only exocrine part secretion, and that is enzyme takes part, right? We are not talking about insulin, which is being secreted by islets of Langerhans, beta cell in islets of Langerhans. So here we are talking about what? We are talking about only the exocrine part, that is secreting enzymes. So what about pancreatic secretion? Talking about pancreatic secretion, we have that it has what? It is the most alkaline secretion. It is the most alkaline secretion. And the most acidic one was what? The most acidic one was our gastric secretion, but here it is the most alkaline secretion, right? Okay. Okay, now what is its pH range? Its pH range is what? 8 to 3, right? 8 to 3. This is its pH. Okay. What we are saying? We are saying pancreatic. We are talking about pancreatic secretion whose pH is 8 to 3 and its nature is alkaline, right? Its nature is alkaline. Okay, cool. Its nature is alkaline. Now Now what we have is, after pancreas we have what? We have bile, right? Bile as a what? Intestinal secretion. Now after bile, our food finally reaches to intestine. And talking about intestinal secretion is also alkaline secretion, right? It is also alkaline secretion, right? Okay. And here, and this is being secreted by Brunner's gland, right, which is present in duodenum, right? Here the pH is 8.8 to 9, 8.9, right? Okay, 8.9. Okay, this is all about briefly different secretions. Now we'll move further more detailedly, right? Moving further detailedly, what we have is, we have the first topic of discussion, and that is salivary secretion. Now talking about salivary secretion, right? Okay, let us move toward, okay, what I said is the most, what is the largest secretion in GI system? We said that is gastric secretion, 2.5 liter per day. Similarly, what about salivary secretion, right? Salivary secretion, talking about saliva, what is the amount of salivary secretion, right? Saliva secretion, it is 1.5 liter per day, 1.5 liter per day. So now what about its pH? It is what? It is, its pH is near to 7, right? Its pH is near to 7. And another fact that we need to know right here is, it is hypotonic to plasma. Hypotonic to plasma, right? It is hypotonic to plasma because what happens is here in this in the area of salivary secretion, there is the permeability for our NaCl, right? Sodium chloride, but it is impermeable to our water. So if NaCl moves out, then obviously there will be the condition of hypotonic, right? So it is hypotonic to plasma. Now talking about what is the source of salivary secretion. Talking about the source, the main source is right here. We have what? We have salivary gland. So this is the main source of saliva, right? Okay, talking about salivary gland, there are two types of salivary gland. One is the major salivary gland and another is minor salivary gland. What are the major salivary gland? These are parotid, submandibular and sublingual. The maximum source of our salivary saliva, right, is from our submandibular. As you can see, it provides about 70% of total saliva. 70%. These are what? These are major, right? Similarly, talking about parotid, it provides 20%. Talking about sublingual, it provides 10% of secretion. So, and these are minor. And under minor, we have von Ebner's gland. What? Von Ebner's gland. So it is so important. We need to know about major and minor. What are the major? The major are parotid, submandibular, sublingual. Submandibular providing the largest concentration or largest amount, and then we have parotid, and then we have sublingual. And what about the minor? Minor, we have von Ebner's gland, right? Von Ebner's gland. So now okay, right here what we have is a little bit more fact about von Ebner's gland. What we can add here is, von Ebner's gland, what? It produces lingual lipase. What I'm saying is, it produces lingual lipase. What this lingual lipase does is, this lingual lipase, it will help in lipid digestion. It will help in lipid digestion. Now, okay, these are the brief introduction about salivary glands. Now moving further, what are the function of salivary gland? Talking about the function of salivary gland, what we have is, it helps in digestion, right? It helps in lubrication and it helps in protection, right? Okay, we're talking about saliva, right? Right now we are talking about salivary gland, right? Okay, now talking about digestion, right? It helps in digestion of what? It helps in digestion of carbohydrate and lipid. As you can see, it helps in digestion of what? Our carbohydrate and lipids, right? Okay, carbohydrate is being digested by salivary amylase. What is this? Salivary amylase. What is salivary amylase also called? It is also called ptyalin, right? And talking about lipid, it is digested by, lipid are digested by lingual lipase, right? And these lingual lipase, as we have mentioned earlier, what we have mentioned, lingual lipase are being produced by our von Ebner's gland, right? So here it is mentioned, it is obtained from Ebner's gland or von Ebner's gland. Now moving further, okay, we said it helps in digestion of carbohydrate and lipid. Just we said only carbohydrate and lipid. We are not mentioning about what? We are not mentioning about protein. So protein digestion will not take place in our mouth, right? So if you will eat anything so purely protein, I mean 100% protein, then you will not have any taste in your mouth, because there will not be digestion of protein at all in our mouth. So protein digestion will not take place. Digestion of carbohydrate and lipid will take place in our mouth. So talking about lubrication, yes, obviously it will help in lubrication of food material, right? And it will be easier to intake. And talking about protection, one good example that I would like to mention right here is, as you can see, the animals, pets, right, cat and dogs, they lick their wounds, right? They lick their wounds. They lick their wounds because while licking, their saliva contains what? Their saliva contains some of the factor that helps that wound in healing. So that is what the protection is. So what saliva contains? The saliva contains, right, secretory IgA, immunoglobulin, right, IgA. Now it contains what? Secretory IgA, that is what? Our immunoglobulin. Similarly, it contains what? Antibacterial factors such as lysozyme and lactoferrins. Lysozymes and lactoferrins. So as I've already mentioned that our saliva does not contain enzyme for protein digestion. It does not contain enzyme for protein digestion. Now moving further, what we have is, we have a regulation of what? Salivary gland, right? How are salivary gland regulated? Our salivary gland, it is so important point right here. Our salivary gland is solely, right, it is only regulated by autonomic nervous system. And we know autonomic nervous system parts, parasympathetic and sympathetic, right? And okay, if you just recall ANS, then we have learned that the parasympathetic and sympathetic nervous system, they act oppositely, but here in the case of salivary gland, they are acting together. I mean their main function is to increase saliva. I mean they both are acting in order to increase the secretion of saliva. So they are not acting opposite to one another, they are acting what? With each other, right, parallelly. So but the secretion, the type of secretion of saliva in different system is different, such as in the case of parasympathetic activation, there will be thin watery saliva. In the case of sympathetic activation, there will be thick saliva. So that is the difference. Now talking about what is this? This is the regulation part, right? Regulation by ANS under two system, parasympathetic and sympathetic. Okay, now let us move toward more detail. So right here you can see innervation, right? Now what will innervate is the cranial number 9, right, the glossopharyngeal nerve. Glossopharyngeal nerve will what? Will innervate, right, will innervate our parotid gland. Now how it will innervate our parotid gland is, it will innervate our parotid gland by what? Through optic ganglion, right, through the innervation of optic ganglion. So innervation, what is the innervation? What is the nerve supply of our salivary gland? Talking particularly about parotid gland, it is being supplied by, it is being innervated by cranial number 9, that is glossopharyngeal nerve, and this glossopharyngeal nerve will be innervating parotid gland with the help of or through optic ganglion. Similarly, talking about what? Submandibular gland and sublingual gland. What? Everything that we are speaking right now, these are the part of major salivary gland, right? Okay, talking about submandibular gland and sublingual gland, what we have is, we have innervation from what? 7th cranial nerve, and that is facial nerve, right? That is facial nerve. Okay, what will happen? This facial nerve will innervate our these glands through submandibular ganglion. So that is about what? Innervation. Okay, now here as you can see, here it is mentioned about the regulation of saliva secretion. Okay, as you can see, the regulation of saliva secreted by parasympathetic nervous system, right? Okay, as you can see, right, what is this? Salivatory nucleus of medulla. It is the main center for what? It is the main center for our saliva secretion. So salivatory nucleus of medulla, where it is how it is activated? It is activated by the pressure in mouth, right? Now how it is inhibited? It is inhibited by sleep, fatigue and fears, right? Okay, Now what will happen is, when we have smell, when we taste, or when we sound, right, when we see food material, at that time what will happen is it will activate our this salivary nucleus of medulla. Now what will happen is, since we know that otic ganglion and submandibular ganglion, right, otic ganglion, it is innervated by our what, glossopharyngeal, and submandibular is innervated by our 7th cranial nerve, facial nerve. So what they will do is they will secrete acetylcholine. As acetylcholine will be released, they will go and act on parotid gland and submandibular gland, and this will result in increased saliva secretion, right, increased saliva secretion. Now increased saliva secretion will also be what, facilitated by acinar okay, it occurs with the help of acinar secretion and through vasodilation, right. Okay, this is the diagram. Okay, now moving further, what we have is we have gastric secretion. Now we already talked about saliva, right, saliva in our mouth. Now just moving below, right? I mean, okay, as I've drunk water, it just reached our mouth, and then after mouth what we have is we have stomach, where it will reach. Now talking about the gastric secretion, right, as we said, what is the total amount of saliva secretion per day? It was 1.5 liter per day, right. Similarly, gastric secretion, we mentioned earlier that gastric secretion is the largest amount of secretion, right, the largest amount of secretion, and that was how much, 2.5 liter per day. And another fact we mentioned is it is the most acidic secretion. Okay, right. And what was the acidic secretion, what was the pH of secretion? It was 1.5 pH, right. Okay, now gastric secretion, right, gastric secretion. Now source of our gastric secretion is what? The source of our gastric secretion is our gastric glands, right. Okay, now what is this gastric gland? Okay, now what will happen is this gastric secretion will take place from certain cells, right. And where are these certain cells found? These certain cells are found in our stomach, right. And in stomach, where particularly these cells are found? These cells are found in our gastric gland. Okay, that means gastric gland contains several cells that help in secretion of gastric secretions. Yeah, that's right. So now talking about the shape of our gastric gland, it is flax shaped. It is flax shaped. Okay, now let us move further. Okay, before getting into the secretion part, let us look toward this diagram, our stomach, right, the gastric region. Okay, here what we have is esophagus, right, and as esophagus ends with the help of lower esophageal sphincter right here, what we have is we have the above, or the highest part, that is fundus, right, fundus, and this area is cardiac, and you can see the body area, right, you can see the antrum, or the antrum of pylorus area right here, right. Okay, right here, as you can see, antrum, it is mentioned, it secretes mucus, pepsinogen and gastrins. We'll look this in detail, right. As you can see, pyloric sphincter is right here, that differentiate what, duodenum and stomach. Similarly, we have body, that is secreting what, mucus, pepsinogen and HCl, right. Okay, that's all. Now moving further, what we have is, yes, this is what, this is our flax shaped what, flax shaped gastric gland, right, flax shaped gastric gland. And what we said is this flax shaped gastric gland consists of different cells, and that help in gastric secretion. Okay, these are the cells, 1, 2, 3, 4, 5, 6, right, these are the cells. Now what about the first cells? As you can see right now, the first cell is our surface cell. As name suggests, why surface cell? As name suggests, because it is present in the surface, right. And we know the fact that our stomach consists of HCl secretion, right. So if stomach contains HCl, then it might erode the wall of what, wall of stomach, but it's not happening like that. Why? Because these surface cells, right, which are present on the surface, they produce mucus. What they're producing is mucus. So what's that important about mucus? It secretes mucus, and the important thing about mucus is that it contains our bicarbonate ion. Okay, now we can relate. How we can relate is, our stomach was containing what? HCl, right? That may erode our stomach lining, but it's not eroding our stomach lining because stomach's surface consists of surface cells, right? Surface cells. Now what these surface cells are doing, they are producing mucus. Now the importance about mucus is it is rich in bicarbonate ion, and as it is rich in bicarbonate ion, it is alkaline in nature and it will cause acid neutralization. That's all. That is the importance of surface cell. So okay, I think you're clear about surface cells. Now moving further, what we have is we have cell number 2, and that is neck cell. Now as name suggests, right, neck cell, it is somewhere present in neck like region. As you can see, it is present somewhat in neck like region. Now what is going on here is, okay, here these neck cells consist stem cells. Okay, I mean to say this neck region consists neck cells, that can be also said as, that can be also called as stem cells. Now what are the importance of stem cells? The importance of stem cells are, it helps in stomach, right, regeneration. It helps in stomach regeneration. Okay, it helps in stomach regeneration. So that is the importance of our neck cells or stem cells. Now moving toward the third number cells, and the third number cells is our parietal cells. Now the importance of parietal cells. Okay, before some time we were talking about HCl present in stomach, right? Now that HCl, how it came into stomach? It came into stomach because of parietal cells. Now what is parietal cell doing? It is producing gastric acid. Now what is gastric acid? Obviously our HCl. One importance of parietal cell is it is producing gastric acid, and another importance is it is producing intrinsic factor of Cashel. Now what is this intrinsic factor of Cashel? It is what? What is its role? Its role is that our what? Vitamin B12, right? You remember vitamin B12? Vitamin B12, right? What is vitamin B12? It is extrinsic factor, right? It is extrinsic factor. Now this extrinsic factor, right, requires what? Intrinsic factor to be absorbed, right? And what that intrinsic factor, now where that intrinsic factor comes from? That intrinsic factor comes from our parietal cell. So that is the importance of our parietal cells. So there is one important clinical condition in which, right, there will be autoantibody against, what I'm saying is autoantibody will be produced against what? Our parietal cells, right? If there will be autoantibody against parietal cells, this autoantibody will destroy these parietal cells. What will happen? There will be no production of gastric acid, and that condition is called what? That condition is called achlorhydria. ACHLORHYDRA. No production of gastric acid or no production of HCl, and that condition is our achlorhydria. And similarly there will be no production of intrinsic factor of Cashel also. So there will be what? There will be no absorption of our what? No absorption of vitamin B12. Now this vitamin B12, what? It will not be absorbed, so our body will what? Will suffer from anemia, right? Anemia. So under anemia, which particular anemia the body will be suffering from? It will be suffering from pernicious anemia. So we talked about different thing about parietal cells. What we said is parietal cells, it helps in production of gastric acid, right? That's HCl. It helps in production of intrinsic factor of Cashel, right? That will help the extrinsic factor or vitamin B12 to be absorbed within our body. So we talked about the condition in which there will be autoantibody formation, right? And if there will be autoantibody formation, there will be no gastric acid production or chlorhydria. Similarly, there will be no intrinsic factor of Cashel, that will not allow the absorption of vitamin B12, and the person will suffer from pernicious anemia. So this was about parietal cells. Now moving further, what we have is we have the number 4 cell. Now what is this number 4 cell? The number 4 cell is our enterochromaffin like cells. What I'm saying is enterochromaffin like cells. Now what is about this enterochromaffin like cell? The enterochromaffin like cell is a source of histamine, right? It is the source of what? It is the source of histamine. So what is the importance of histamine right here? It is the source of histamine that will take part in what? That will take part in enhancement of production of our acid or gastric secretion. So that is the importance. So if you recall the pharmacological fact that in the case of what peptic ulcer, what we were doing is we were providing what we were supplying, or we were prescribing ranitidine and cimetidine, right? What are these ranitidine and cimetidine? Ranitidine and cimetidine, these are antihistaminic, right? These are antihistaminic. What they will do is they will not allow, they will what, they will antagonize this histamine, and if this histamine will be antagonized, then will it be able to increase gastric secretion? No, it will suppress the gastric secretion, and that's what we want in peptic ulcer case. So what we have is what we discussed right now, enterochromaffin like cell, it helps in secretion of histamine, right? That will enhance enhance gastric secretion or HCl secretion. And we mentioned the example of ranitidine and cimetidine. What these ranitidine and cimetidine are doing, these drugs, these are antagonizing histamine, right? And if the these are histamine receptor blocker, right? These are histamine receptor blocker. It will not allow histamine to act on its receptor, because of which it can't increase the acid secretion. It will cause what? It will cause the decrease in acid, right? And that's what we want in the case of peptic ulcer. So moving further, what we have is we have cell number 5. Now what is this cell number 5 doing? It is, what is the name of cell number 5? It is chip cell. Now what is this chip cell doing? It is producing pepsinogen, and you know the pepsinogen, it will be converted to pepsin for protein digestion, right? This was all about chip cell, right? Chip cell, it will produce pepsinogen. If anything ends with gen, that means it is what? It is inactive enzyme. So now moving further, what we have is we have the last cell, and the last cell is our cell number 6, and that's G cell. Now these G cell are present in the region of antrum. Now what is this antrum? Antrum, it is present in the pylorus region, right? In pylorus region, we can say pylorus as pylorus of antrum also. So these antrum, what these G cell are doing, as name suggests, G, these are producing gastrin, right? Gastrin, okay, these are secreting gastrin. Now we will learn about these gastrins, right? This will learn about these gastrins and other different what hormones present in our this GI system in hormonal secretion part. So right now only gastrin. Okay, now okay, this was all about the different cells right here, right? Okay, now what we have to do is we have to just look in detail about our parietal cells, because it is producing what? HCl, right? Gastric acid. Now talking about parietal cells, right? Okay, let us just look at the diagram mentioned in our guidons, right? Okay, right now we can see what? We can see this is our mucous layer right here, right? You can see the surface mucosal cells right here. What are these? Surface mucosal cells. And what surface mucosal cells were doing? They were producing bicarbonate, right? They were secreting bicarbonate. And another important thing I forgot to mention is these are also what? These are also secreting trefoil peptide, and trefoil peptide, right? Trefoil peptide. And the importance of trefoil peptide is it helps in what? It helps in protection. And bicarbonate secretion, we already said, it helps in what? It helps in what? It helps in our acid neutralization. Moving further, we have mucous neck cell and neck cells. We mentioned neck cells. Now what these neck cells are doing? These are stem cells that are helping in regeneration of stomach, right? Similarly, moving further, we have parietal cells. What these parietal cells are doing? These are producing acid, gastric secretion, and acid, gastric acid, right? HCl. And these are also helping in intrinsic factor secretion, so that we can absorb extrinsic factor, that is vitamin B12. And another cell we mentioned is enterochromaffin like cell. Now what these enterochromaffin cells doing? These are secreting histamine. Similarly, we have chip cell that is secreting pepsinogen, that will be converted to pepsin for protein digestion. So this was all about what? Different cell in gastric gland. Okay, now what we have right here is we have mentioned about here. This diagram helps to show us about the regulation of gastric acid. So right here, if you see, talking about the regulation of gastric acid, right? Okay, as you can see, what is this? This is our antrum, this is our fundus, right? Antrum and fundus. Okay, we have antrum region right here, fundus right here. What is mentioned right here is gastrin is released from G cell. As you can see, what is this? This is our G cell. What is being released from gastrin is, what is being released from our G cell is our gastrin, right? Our gastrin. Now this gastrin, right, this is happening in the region of antrum, right, on the response of gastrin releasing peptide, right? What is this? Gastrin releasing peptide, right? Gastrin releasing peptide will cause G cell to secrete gastrin. Now what will happen is these gastrin will travel, right? Where they will travel is this gastrin will travel through the circulation, right? Now where they are going is they are going into enterochromaffin like cells, right? Enterochromaffin like cells. And another what we have is, we have one is enterochromaffin like cell right directly right here, and another is our parietal cells. So as you can see, right, okay, as you can see, travel through the circulation to influence the activity of enterochromaffin like cells, right, and parietal cells. Now ECL, enterochromaffin like cells, will release what? This will release histamine. As we know, enterochromaffin cells will release histamine, which now this histamine also will act in parietal cell, right? Act in parietal cell. Okay, now what is going till now? What happened is our this GRP, it acted on our G cell. Now G cell will release gastrin. This gastrin will move through circulation and it will go and it will act on ECL, enterochromaffin like cell. And another thing is it will act on parietal cells. Now on acting on ECL, it will release histamine, and that histamine will again what? It will go and act on parietal cells. Okay, till now this was the story. Now acetylcholine, right? Acetylcholine released from nerves is an agonist for ECL cells. So now this acetylcholine, as you can see, this is okay, this is our acetylcholine and it is being produced by nerve ending. Okay, this is our nerve ending, this is acetylcholine. This acetylcholine will also act as agonist for our ECL cells, right? Not only ECL cells, it will act agonist for our chief cell and parietal cells, right? Okay, so this is somewhat about the regulation. We'll just go a detail later on, but this is the diagram mentioned on Guyton. So here we came to know about some of the idea. So right now what is mentioned right here is the parietal cell produce intrinsic factor of Castle, as we have known, and it will help in vitamin B12 absorption. Yes, we know it, we discussed it earlier. Autoantibodies to parietal cells, what it will happen is there will be anti-parietal cells antibodies, right? It will not allow to form HCl, and that condition is echlorhydria, and it will not allow extrinsic factor or vitamin B12 to be absorbed, and that condition is pernicious anemia. Okay, so as I had mentioned earlier that we were going to see about what we are going to have a look about these parietal cells. Now right now this is our parietal cells, right? This is our parietal cells. Now looking toward parietal cells, as you can see, our parietal cells, what is going on? What is our parietal? This is our parietal cells. These parietal cells at the one end, right? One end, it is near to blood vessels, right? It is near to our these blood vessels. And another end where it is, and another end, this is what? This is our luminal, this surface, and the surface it is close to is our luminal surface. Right now what we discussed is one end of our what? Parietal cells. One end of parietal cells is close to blood vessel and another end is close to luminal cell, luminal surface. Now what is going on is, okay, what is going on right now is because of metabolism, right? Because of metabolism within parietal cells, there will be the production of large amount of CO2, right? Large amount of CO2, as you can see right here, CO2. Now this CO2 will bind with our water and it will produce carbonic acid, H2CO3, carbonic acid. Now this carbonic acid, right, this carbonic acid will be broken down into bicarbonate ion and H plus ion. Bicarbonate ion and H plus ion. Now which enzyme will take part here? It is carbonic anhydrase, obviously. Now what will happen is this HCO3 or bicarbonate ion and this our proton or H plus ion. Let us talk about this proton, right? This proton will be carried to our luminal surface, as you can see right here, right? And as it is going outward, it is being balanced by our potassium ion by carrying inside or by coming inside. Now this pump that will help in outward going of this proton and inward bringing up our potassium is called proton pump, right? Proton pump. Okay. Now this is the story of our H plus. Now what about our HCO3? Now bicarbonate ion, what will happen is this bicarbonate ion, as it will be produced in parietal cells, it will be carried to our blood vessels. This bicarbonate ion will be carried to our blood vessels, right? Okay, that means as our H plus ion moves out toward luminal surface, our this HCO3 ion will move toward our what? Blood vessels. Now here is one pump, and that is our what? As the HCO3 or bicarbonate ion moves outward, it will be balanced by Cl minus ion being carried inward, right? And this is being facilitated by chloride bicarbonate exchanger. So now this chloride ion, which is inside the parietal cell, will be carried outward, right? See, now since there was already the presence of H plus ion, right? Now what is going on is there is now chloride ion also right here. So by the fusion of these two, there will be the production of HCl. So this is what what our parietal cell, this is what is happening on parietal cells. Now as you can see, there is one important point that is being written here. For every HCl produced, HCO3 ion is pushed into the blood. That means for every HCl being produced, means what? For every proton being what? Pumped into luminal surface, or for every Cl minus ion being brought into luminal surface, there will be what? HCO3 ion which is being carried to blood vessels, pushed into blood or blood vessel. So this is the fact. Okay, now let us move toward another thing. Okay, right now what we can mention right here is we can mention that as the proton pump will be more active, there will be the more production of HCl, right? What we are saying is, as there will be the more activity of proton pump, there will be the more production of HCl, and that's right? Okay, that's right. Now let us see the different condition in which there will be the activation or the increase in activity of proton pump. Right? Talking about the different point, right? The first thing is, the most potent thing that will stimulate the proton pump is histamine, as we discussed earlier, right? Enterochromaffin like cells will produce what? Histamine. That will increase what? That will increase HCl production, we said. Now how it is increasing HCl secretion? By what? By increasing the activity of our proton pump. That is a fact. And another thing that will increase the activity of proton pump is our gastrin, right? Gastrin. Now what is this gastrin, right? What is this gastrin? Gastrin is what? Hormone that is being produced by G cell. If you remember, we talked in what? Gastric glands, G cells. Now this gastrin is mediated through what? This is mediated through CCK beta receptor. It is important when we will be discussing about the hormones, right? Gastrin, right? Gastrin will be mediated through CCK beta receptor. Similarly, another thing that will increase the activity of this proton pump is our acetylcholine, right? And how this acetylcholine will be activated? It will be activated through M3 receptor. So now there is one important concept, as I've already mentioned. What? As I've already mentioned that for every acid being produced, for every HCl being produced, there will be one bicarbonate ion that will be moving into our what? Vessels. So now this effect is called whenever we'll have food, right? Whenever we'll have food, what will happen is whenever we will have food, there will be what? There will be HCl production. What I'm saying is, whenever we'll have food, there will be what? There will be HCl production. HCl production, right? Now, as HCl will be produced, right? As HCl will be produced, what will happen is, as we mentioned, that for every HCl produced, HCO3 ion will be pushed into blood. Now, as HCl will be produced, what will happen is, our blood vessel will contain what? Our blood vessel will contain HCO3 minus ion, right? And we know the nature of this HCO3 minus ion is alkaline, right? There will be what? Alkaline condition in our blood vessels, right? So what we can say is, after having food means, we can say that it is the condition of postprandial, right? Postprandial, that means after food intake. Now postprandially, what is going on is, one HCO3 is moving into our, for every HCl produced, one HCO3 is being what? It is being taken to blood vessel. So postprandially, there is the alkaline condition that is being found in our blood vessel, right? Alkaline condition. So this process, right, whenever we take food, right, after food intake, right, or postprandially, the blood vessel will have what? Alkaline condition or high level of bicarbonate ion, right? So this condition is called our postprandial alkaline tide. How this name came? Postprandial. Why postprandial? Because it is happening after the food intake. Alkaline. Why alkaline? Because there is the high accumulation of bicarbonate ion into blood vessels, or there will be the alkaline condition. That's why. So this is one important condition, right? This is one important concept that we need to know. Now, as we are into parietal cells, right? As we are into parietal cells now, okay, as I have said earlier, after the intake of food, what will happen is increase in HCl production, right? As the HCl production will be increased, there will be the increase of HCO3 ion into the blood, right? And since there will be the increase of HCO3 ion into the blood, this is what? Postprandial alkaline tide condition. So that's all. Now, as we have mentioned right here, parietal cell, this is also the same diagram, but it is from what? Guytons. So I would like to focus right here. So as there will be the increase in metabolism, right, where the metabolism will be increased into the parietal cell, there will be the high production of CO2. There will be high production of what? CO2, right? CO2. Now this CO2 will combine with H2 and it will form what? Sodium bicarbonate, and that sodium bicarbonate will be what? Acted by carbonic anhydrase, and it will be there will be the formation of H plus and HCO3. And we have mentioned, right, what is going on is this, as you can see, this is our lumen, right? And what we have here is we have our bloodstream, right? Lumen, right? This is our lumen. Okay, now what is going on is this H plus, right, it will be pumped out, right? It will be pumped out right here. Now as it will be pumped out, our potassium will be pumped in, and it will take place through what? Sodium potassium ATPase, or this is called what? Our proton pump. Similarly, what is going on is this bicarbonate ion, it was what? It was being carried out, right? It was being carried out, or you can see right here, where you can see is, we can see right here, this bicarbonate ion, it is being carried out, right? It is being carried out, and chlorine ion is being carried in, right? Carried in. As chloride ion is carried in, it will be what? It will follow this path and it will be taken out, right, into the luminal surface, and H plus and Cl minus, right, here they will combine to form HCl. So that's all, right? Okay, this is all about parietal cells. Now moving further, what we have is, we are going to talk about agent, right, altering acid secretion or changing acidic secretion. So what we have right here is, we have what? We have the factor that will increase acid secretion, and we have the factor that will decrease acid secretion. So talking about the factor that will increase acid secretion, we have is histamine. Yes, we have talked earlier that histamine is the most potent factor for increasing proton pump. Yes, we talked about gastrin, right, which was mediating through CCK beta receptor. Yes, we talked about this also. We talked about acetylcholine that was acting through M3 receptor. That's all. And the factor talking about the factor that will cause decrease in acid secretion, what we have is, we have acid itself. That means more the HCl, right, it will cause the decrease in HCl. This is what? Negative feedback mechanism. That's all. Excess acid will cause the decrease in acid secretion. Similarly, talking about another we have somatostatin. Now what is the somatostatin? It is universal inhibitor. Universal inhibitor, right? This is causing what? This is causing the inhibition of production of acid secretion. Similarly, we have prostaglandins, right? We have prostaglandins. Now this prostaglandin will also cause the inhibition of acid secretion, right? And as you know, the NSAIDs, what are these NSAIDs? These are the prostaglandin blocker. What they will do is, since the prostaglandin was what? It was prostaglandin, if you remember, right? Prostaglandin, it is what? It is facilitating to produce mucus, right? If you remember mucus, right? And prostaglandin, what? Prostaglandin, okay, NSAIDs, we're talking about NSAIDs. What these NSAIDs were doing, these NSAIDs will what? These are NSAIDs are prostaglandin blocker, and we said that prostaglandin will inhibit the production of acid. Similarly, our this NSAIDs, what they are doing, they are causing the increase. They are what? They are prostaglandin blocker. What they will do is, they will cause what? Increase in acid secretion or decrease in acid secretion. They will cause increase in acid secretion, and that may result the peptic ulcer. So NSAIDs induce peptic ulcer, right? Okay, that's all. Okay, as you can see, as I was saying earlier, prostaglandin, that what the prostaglandin are doing, they are causing what? They are causing mucus production, right? Mucus production, right? Mucus production. And what these mucus are doing, they are producing bicarbonate ion. What these bicarbonate are doing, they are what? They are acid neutralizing, or they are decreasing HCl level, right? So prostaglandin, they are facilitating the decrease in acid secretion. But if something will come that will what? That will block these prostaglandin, then obviously they will induce what? They will induce the production of HCl, and that will ultimately cause the production of, that will ultimately cause the result of peptic ulcer. So this was all about what? Agents that was altering the gastric secretion. Now the role of gastric secretion. Now talking about the role of gastric secretion, what we have is, talking about the role of gastric secretion, we have is the gastric secretion help in digestion, right? Digestion. Now talking specifically, it will help in digestion of lipid, and it will help in digestion of proteins, right? And it will not help in digestion of carbohydrate, right? It will not digest carbs, right? Okay, talking about the lipids, there is our gastric lipase. If you remember, there was lingual lipase that was being produced by von Ebner's gland, right? That was digesting lipids in our mouth. So here we have is gastric lipase. Similarly, to digest protein, we have pepsinogen. As I mentioned earlier, if there will be something gene, then that will be what? Inactive form. So this pepsinogen, it will what? It will be converted to pepsin, right? It will be converted to pepsin, and with the help of HCl, and this pepsin will help in protein digestion, and this is what? This is main thing for protein digestion. But in the case of cow, right, in the case of animals like cow, we have renin, right? They have renin. Now these are also responsible for protein digestion. So we said that lipid and protein are being digested in the stomach, but no carbohydrate, right? No carbohydrate. Now talking about the gastric secretion phase. Okay, now we will be talking about gastric secretion phase. Gastric secretion phase. There are how many? There are our 3, 1, 2 and 3. The first one is cephalic phase. Now what is the condition? What is the phase? I mean, what will happen in this cephalic phase is there will be absence of food. What do you mean by absence of food? That means we will just hear about food, we will just see food. That is the condition, right? Whenever we see food, at that time there will be water in our mouth, right? And that is saliva. That is what the cephalic phase is. It means there is absence of food, but we are hearing about food. We are having sight of food, right? Okay. And in the absence of food, right? And this is responsible for 20% of acid secretion, right? 20% of acid secretion, and we're talking about what? Gastric, HCl. Similarly, gastric phase is what? The phase in which there will be food inside our what? Stomach, right? Stomach. And that is responsible for the major acid secretion, and that is 60 to 70% of acid secretion, and this is also considered to be the major phase. Okay, now as the food will leave stomach and it will enter into what? Intestine. At that time what will happen is there will be intestinal phase when food moves to intestine, right? and this will be responsible for 10% of acid secretion. Okay, this was all about what? This was all about our gastric secretion. Now moving into another topic, that is pancreatic secretion. And under this pancreatic secretion, initially we need to know that there are two parts of pancreas. One is endocrine part, right? Endocrine cells that will help in the production of what? Hormones. And another is exocrine cells that will help in secretion of enzymes, right? And in this chapter we'll be focusing about the enzyme part. That means the exocrine part of pancreas. So here as you can see right here we are talking about exclusively exocrine part, right? The part that will secrete what? That will secrete enzymes. Talking about the pH of our pancreatic secretion, it is 8.3, alkaline, right? Alkaline in nature. And what is this pancreas doing? It is digesting carbohydrate, lipid, protein, all three things. Now this carbohydrate is being digested with the help of pancreatic amylase. If you remember salivary amylase in our saliva in the case of our mouth. Similarly, lipid, we have pancreatic lipase, right? And this pancreatic lipase is assisted by cholipase for lipid digestion. Similarly, in the case of proteins, we have trypsinogen and procarboxypeptidase. I have said earlier, gen means inactive, and if something will be in front pro, then that will be also inactive, right? Okay, now what these things are called? These things are called zymogen, and this zymogen means inactive. That means in a nutshell we can say that trypsinogen and procarboxypeptidase, these are called zymogen, right? Yeah, that's right. And what is the meaning of zymogen? That means inactive, right? Now why these are present in inactive states? That means why these are present in inactive state in pancreas? Because if it will be present in active state, then there may be what? There may be the auto digestion, right? So to prevent that auto digestion, they are present in inactive form. So to prevent auto digestion of pancreas, so how they will be converted into the active form? They will be converted into active form in small intestine, and in small intestine what we have is we have enterokinase, and this enterokinase, it will convert this trypsinogen which is in inactive form into trypsin, that is the active form. So this is how it is going, right? Okay, now talking about the regulation of pancreatic secretion, the hormonal regulation, it is being regulated by two hormones, and they are cholecystokinin, pancreozyme, and secretin, right? Okay, now focusing on cholecystokinin. Now cholecystokinin simply means contraction, anything that helps in contraction of gallbladder, right? Cholecystokinin is what? It helps in gallbladder contraction. So right here, if you see right here, okay, cholecystokinin, pancreozyme. So what is going on here is it is enhancing pancreatic secretion. This is enhancing pancreatic secretion which is rich in digestive enzyme. As you can see, enzyme, pancreozyme, right? Zyme. It is enhancing pancreatic secretion, right? Which is rich in what? Enzyme, digestive enzyme. So, and cholecystokinin, as we have already mentioned, what? It will help in contraction of gallbladder, that will help to release bile, right? That's all. What about secretin? Secretin will enhance pancreatic secretion, but that will be not rich in what? Digestive enzyme. That will be rich in HCO3 minus. So what is the difference between these two regulating hormones? The difference between these two regulating hormone is that cholecystokinin, pancreozyme will enhance pancreatic secretion, but that will be rich in what? That will be rich in our digestive enzyme. But in the case of secretin, it will enhance pancreatic secretion, but it will be rich in what? HCO3 minus, right? That's all. Another thing is cholecystokinin help in contraction of gallbladder, and that will helps to release bile. So okay, what we said is, as it is mentioned, most potent chloragouge, and chloragouge is the one that will stimulate the gastric that will stimulate the contraction of the contraction of our gallbladder to release bile, and it contracts the gallbladder to release bile. It is mentioned right here. And secretin, right it will increase pancreatic secretion, but that will be poor in digestive enzyme, or that will be rich in bicarbonate ion, right? And this bicarbonate ion will help in acid neutralization. So that is all about pancreatic secretion. Now moving into bile secretion. Bile, about 500 ml of bile is produced daily. about 500 ml of bile is produced daily, and its pH is about 7.8 pH. And the main source of bile is our liver. And what is going on is, in the case of liver, when the bile is present in liver, it will contain about 97% of water. Okay, now later on, as it will be stored in gallbladder, and there is one important question: where is bile stored? It is stored in gallbladder, and while being stored in gallbladder, there will be the concentration of bile, and at the time of concentration, there will be the loss of water, and the concentration of water now becomes 87%. Here the concentration of water was 97, but here the concentration of water is 87%. So what we can say is, storage and concentration of bile will take place in gallbladder, whereas the production of bile will take place in liver. So this was all about talking about the bile. Bile contains of bile acids, bile salts, bile pigment, and cholesterol, right? Okay, cholesterol. Now here what we can say is, there are, talking about bile acid, there are two type of bile acid. One is primary bile acid and another is secondary bile acid. Now what are these primary bile acid? The primary bile acid are cholic acid, chenodeoxycholic acid, right? Now when these primary bile acid will be acted by intestinal bacteria, that will be converted into secondary bile acid, and they are deoxycholic acid, lithocholic acid, right? That's all. Now another thing that need to be mentioned right here is, conjugation of bile acid with glycine and taurine occurs in liver, and that is what the product glycocholic acid will be formed, glycocholic acid. Similarly, the bile salt, right? What are these bile salt? These are the salt of potassium and sodium. Example, sodium glycocholate, right? Sodium glycocholate. Now talking about the enterohepatic circulation, what we have is, bile is being produced in liver, right? Now on being absorbed, the bile salt is being absorbed, and they are what? They are being carried to the intestine, right? The specific part is the terminal ileum. So there will be the recycling of bile salt for about 6 to 8 times per day, right? Now why the recycling occurs? To reduce the demand to produce new bile salts. So this was all about what? Bile salt, right? Bile secretion. Talking about the intestinal secretion, what we have is, these are the intestinal secretion, right? Okay, talking about the intestinal secretion, what we have is, these are released by crypt of liver coon, right? Now these crypt of liver coon, they contain cells, right? As you can see, 1, 2, 3 cells. Now what are these cells? These are goblet cells, right, or surface cells. What these are doing? These are producing mucus, as mentioned in gastric gland also, bicarbonate rich mucus. Similarly, epithelial cells, right? What are these epithelial cells? These epithelial cells are also called intestinal epithelial cells, right? And these epithelial cells are responsible for small intestine secretion, succus entericus. Okay, talking about the last cell, and that is Paneth cell, right? These are the base cells present at the base, and what they are doing is, right, these are present on the base of crypt of liver coon, and these are producing antibacterial in nature products, such as these increase the concentration of zinc, and another is these are what? These are defensive in nature, that are lysozymes. So this was all about what? This was all about intestinal secretion. So we talked about all these things. So this much for this lecture. So have a great time. Hope to see you in next lecture.